So, good morning, good morning, good morning, good morning. I was sorry for half an hour late class. Uh, it was last night. I was working quite late, 3 a.m. in the morning, and I just woke up at 7:15. So we are starting at 7:30 now. So we will take this class till 8:30. Yeah. So so we are done with yesterday with the basics of your enzyme action. And then we have decided to go for classifications, nomenclature, allosteric enzyme, inhibitors of enzyme, uh, the science behind these enzymes. And now today we're going to talk about little about kinetics of these enzymes, how the kinetic of these enzymes works out. So enzyme kinetics. So little bit of refreshment, little bit of revision. We know this enzyme substrate complex formation in the step 1 of the enzyme working. So first step, enzyme and substrate combines to form a complex, right? And then ES complex is made. Then ES complex further makes your enzyme product complex EP. In between there's a transition state. Then once the EPs went through, then you have your enzyme and product. So product is made and enzyme is ready for another substrate. So this way reaction continues. You can see all these reactions to be reversible. So all these reactions in the step 1, step 2 and step 3, they are all reversible. So substrate concentration and reaction rate, how they are co-interrelated with each other. So here we can see that the maximum, so this is a rate reaction on the y-axis, yeah, and on this on the x-axis the concentration of a substrate S and V. So initially you will have like lots, not so much of substrate concentration, but you have a lot of enzyme there. The enzyme concentration is always there. But in the middle, you will have your all enzyme to be attached to your substrate. The reaction rate will be the highest here. But at one point, it will reach to saturation point that and no more substrate could be attached to it. So this enzyme is saturated for far more substrate that it can deal with. So that means all of enzyme is combined the substrates. So in this case, you get a hyperbolic curve here. And this hyperbolic curve was actually found out by Michaelis and Menton, both two different scientists, and together they have discovered one equation also. So the rate of reaction increases as the substrate concentration increases, and maximum activity occurs when the enzyme is saturated. In biology, the active site is a region of the enzyme where substrate molecules bind and undergo chemical reaction. So the active site consists of residues from the temporary bonds with substrate and residues that catalyze the reaction of the substrate. So although the active site is small related to the whole volume of enzyme. It is the most important part of the enzyme as it directly catalyzes the chemical reaction. It usually consists 3 to 4 amino acids, while the other amino acids within the protein required to maintain the protein tertiary structure of the enzyme. So, what are the units of enzyme activity? So enzymes are expressed in terms of their concentrations, but are not like in terms of the concentrations, but in the terms of the activity, how much they are producing the product from the substrate. So in that sense, there's a classical unit that is unit of enzyme activity, micromole substrate transformed per minute, or specific activity would be micromole substrate per minute milligram enzyme. Molecular activity would be micromole substrate per minute minus micromole of enzyme concentration, and so on. So basically, the activity, if we see the international system of unit, your enzyme unit would be a catal. It is designated as a catal, CKATAL, cat. And in the decimal form, if it is microcatal, that means it's a 10 to the minus 6 catal. And micromolar, it is equivalent to the micromole per second. Micro, or we can also say concentration of enzyme activity as microcatal per liter. Okay. So there are two types: specific catalytic activity, molar catalytic activity. The specific catalytic activity, it deals with the activity of enzyme preparation in relation to the protein concentration. Whereas the molar catalytic activity, this is determined when the pure enzyme with known molecular weight is available. So it is expressed as catal per molecule when the enzyme has only one active site or center per molecule. So the molar catalytic activity equals the turnover number. So reaction rate is expressed as a change in the concentration per unit time. So this we have just discussed now. So there's a specific activity of enzyme in which your unit of activity, enzyme activity per milligram protein is identified. So one unit will be equivalent to the enzyme of enzyme that is able to convert micromole of substrate to product in 1 minute at given temperature and a given pH. And that will be a specific activity. Then there's a turnover number. That's another new term here, which is very important. So turnover number is also termed as K catal, which is defined as the maximum number of conversion of substrate molecules per second that a single catalytic site will execute for a given enzyme concentration. So if it is K catal is equivalent to the Vmax enzyme concentration. That is, for example, carbonic anhydrase, there is an enzyme which has a turnover number of around 600,000 per second. The meaning of that turnover number is that this carbonic anhydrase enzyme is able to change your 600,000 molecules of products per second. That is your bicarbonate products. So it's able to produce this much of amount of products per minute within. Yeah. So that will be your turnover number. So for example, so enzyme has 1.84 gram of weight and the molecular weight is 36,800. So in presence of excess substrate catalyzed at the rate of 4.2 mole per substrate per minute, calculate the turnover number. So the molecular enzyme is equivalent to the 1.84 gram divided by 36,800 gram per mole. So the 5 into 10 is to the minus 5 mole enzyme. So turnover number would be Vmax divided by enzyme. So this will be your 4.2 divided by 5 multiplied by 10 is to the minus 5 that you have identified. Vmax is given here 4.2. So 84,000 per minute is your turnover number. So some of the famous enzymes: catalase, carbonic anhydrase, acetylcholinesterase, chymotrypsin, lysozymes. There all are various different enzymes has their respective functions, and their turnover numbers are also identified. 4 into 10 raised to the power 7, 1 into 10 raised to the power 6, 1.4 into 10 raised to the power 4, and so on. So what does K catal means actually? So K catal is the first order rate constant describing enzyme substrate giving rise to enzyme plus product. So which also known as the turnover number because it describes the number of reactions that a molecule of an enzyme can catalyze per second under optimal condition. So most enzymes have K catal values between 10 raised to the power 2 and 10 raised to the power 3 per second. So enzyme plus substrate give rise to enzyme substrate, and yeah, enzyme plus product. So here uh, this so all different substrates will have different um, rate of reaction this product is producing. So according to that, this is a hyperbolic saturation kinetics for enzyme was identified with relation between the substrate concentration and velocity of reaction. So this is the rate of reaction on the y-axis, and on the x-axis the substrate concentration, right? So what is happening is on the substrate on the x-axis you have a Km, right here, and on the left hand side is half Vmax to be present here. That is your half rate of reaction Vmax, and the maximum is the Vmax on the top, on which the saturation point is attached. So this is the constant, Michaelis constant, and so on. So Michaelis-Menten equation, V naught is equal to Vmax multiplied by substrate concentration divided by Michaelis constant plus substrate concentration. So V naught is the velocity of the reaction at particular point, Vmax is the maximum velocity of the reaction, S is your substrate concentration, whereas Km is your Michaelis constant. Yeah. Km is your Michaelis constant, S is your substrate concentration, and Vmax is the maximum velocity of reaction, whereas the V naught is the velocity of reaction itself. Then Km by and Vmax. So Km, it's a ratio of rate constant, that is K2 plus K3 divided by K1, right? So that's we have discussed in the previous one. So the Michaelis-Menten equation is equal to K1, this is K2 and this is K3. So it's a ratio of K2 plus K3 divided by K1. And it is equivalent to the substrate when initial rate is equivalent to the half Vmax. So it's a property of ES complex which does not depend on the concentration of E or S. So maximum velocity at a fixed enzyme, Vmax is the maximum velocity at fixed enzyme concentration, and it is directly proportional to the enzyme also. So there's one problem. These are the scientists Leonard Michaelis and Maud Menten. So in 1875, 1949, and 1879 to 1960. The problem is that Vmax is never achieved. Is approached but never achieved. So it is going to the top, but still your substrate concentration cannot attach to the Vmax. Then Lineweaver-Burk double reciprocal plot. So it was very hard to identify all these values from that equation, from the Michaelis-Menten equation. Then Dean Burke and Hans Lineweaver, they worked on that equation. What they did first, they reciprocate that equation, they inverted it. So 1 by V naught is equal to Km plus S divided by Vmax multiplied by substrate concentration, and then they divided these vectors, Km by Vmax multiplied by substrate and S by Vmax multiplied by substrate. And then after that you simplify this equation. 1 by V naught equals to Km by Vmax by substrate, and 1 by S, and S is cut it out, and you have Km by Vmax upon S and 1 by V naught. So that's the reaction, how it is done. So this reaction resembles the equation of straight line, that is this will be your Y, and this will be your M, substrate concentration, this will be your X, and this will be your intercept, that is C. So Y is equal to mx, this is your slope, Yeah, this 1 by s will be your x-axis and 1 by mu x will be your intercept. So y is equal to 1 by v naught, m is equal to km by v max, x is equal to 1 by s, and x, the intercept, would be a 1 by v max. So this way, this is c actually. So this is, and this we can draw over the graph. So y-axis 1 by v naught, substrate is 1 by s naught, and then you draw your slope, that is your km by v max, and your intercept here would be your, on the y-axis, 1 by v max. On the x-axis it will be a minus 1 by km here. So we have to, now we want to discuss about enzyme kinetics of various inhibitors that we discussed yesterday, competitive, reversible and irreversible. So we will discuss how they are play a role in the case of a Lineweaver-Burk double reciprocal plot. Yeah, so we will discuss that part now. So why we study inhibition of enzyme activity? There are two reasons. So we can use this inhibition as a probe for understanding the kinetics and properties of enzyme in their uninhibited state. And many, perhaps most drugs are inhibitors of specific enzymes. So the concept of inhibition, it's actually a biological catalyst that alters the rate of reaction without itself becoming altered by its participation in the reaction. So the ability of enzyme to catalyze a reaction can be altered by binding small molecules to it, sometimes at its active site, sometimes at site different from the active site. So they are also inhibitors and accelerators. Inhibitors which tend to reduce the enzyme ability to accelerate the reaction, which is less commonly found, and there's alteration that give rise to increase in enzyme ability to accelerate the reaction. They are accelerators. So why more inhibitors than accelerators? So natural selection, if they have a small molecules that can facilitate the enzyme propensity to speed up a reaction, so nature probably would have found a way to incorporate those into the over the years, and most enzymes are already fairly close to the optimal in their properties, so we can readily match them with the factors also. So types of inhibitors: reversible and reversible. So reversible, they are the one which binds without possibility of release, and they are covalently attached, very tightly attached, and each inhibition event effectively removes a molecule of enzyme from the availability. And the reversible, usually attached by weak non-covalent interaction. They are of several kinds, and classification somewhat superseded by the enzyme structure-based knowledge of mechanisms, but not entirely. Then there's a types of reversible inhibition. There's a competitive, non-competitive, uncompetitive, and mixed. So according to the reversible inhibition, the first, as a competitive one, they're the inhibitor that binds at the active site, which prevents binding of substrate. Then there is a non-competitive inhibitor that binds distance from the active site and interferes with the turnover number. And there is uncompetitive, which is rare. Inhibitors binds to ES complex, removes ES, interferes with the turnover number. Then they are mixed, usually competitive plus non-competitive. So first is your competitive inhibition. So in this, as we know that your substrate the rate will increase, the inhibitor will be outcompeted by the increased number of substrate concentration. So here your substrate will be competing your inhibitor will be competing the substrate activity side. So the kinetics of this reversible competitive inhibitor would be, the competitive inhibitor hinders the binding of substrate but not reaction velocity. So it will affect the Michaelis-Menten constant but not the Vmax. So your speed of your connection will be not changed, but the constant will be changed. So Km is the amount of substrate that needs to be present to run the reaction velocity up to half its saturation velocity. Whereas the competitive inhibitor requires us to show more substrate into the reaction in order to achieve that half maximum velocity. So competitive inhibitor, it increases Km. So competitive inhibitor, so Km goes up to minus 1 by Km, move towards the region. So here we can see in the competitive inhibition, as we suggested, the Vmax does not change, but your Km is changing, right? So as in the normal case, this is the normal case, the first line here. This is an uninhibited enzyme, and then with a low concentration, and then further high concentration. Now uninhibited, low concentration, and then high concentration. So during this time, if the slope is changing, but your Vmax is not changing. It's independent of inhibitor concentration. But your Km is getting decreased and it's going near to the Km actually increasing, but according to the graph, if it is minus 1 by Km, 1 Km, it will move towards the origin. And then comes non-competitive inhibition, in which inhibitor does not attach to the substrate activity site but to the another one. So non-competitive inhibitor has no influence on how available the binding site for the substrate, so it does not affect the Km at all. But however, its profound inhibitory influence on the speed of reaction, that is its turnover number. So it reduces Vmax and has no influence on Km. So we have these non-competitives which decrease in Vmax and 1 by Vmax is increased. So which x-intercept is unaffected. So in this one, decrease in Vmax is equal to 1 by Vmax is increased. So x-intercept is unaffected. So have your uninhibited enzyme here, and these are the enzyme with the low concentration and enzyme with the high concentration. Yeah, this and your Vmax is uninhibited here, and your yeah. So, but your Km is unaffected in this non-competitive inhibitor. So they will come and join at the one point. They will not change, but your Vmax will be keep on going up. So the Vmax value will be decreased, but 1 by Vmax will be increasing on top. Then there is the uncompetitive inhibition. So we have discussed three types so far: competitive inhibitor, non-competitive inhibitor. Now third one is your uncompetitive inhibition. So in this one, the inhibitor binds only if enzyme substrate has already formed. So it's create a ternary ESI complex. So it will attach to the enzyme substrate complex in order to make your ESI complex. So this removes ES by Lee-Chatelier principle. It actually drives the original reaction. E plus S gives rise to ES to the right. So it decreases Km. But it interferes with the turnover, so Vmax goes down. If Km and Vmax decreases at the same time, then it's classically uncompetitive inhibition. So this is the graph for uncompetitive inhibitor. So Km moved towards origin, and where Vmax moved away from the origin. So you have your slope here, Km by Vmax, which is unchanged. Your slope is not changing, but your Vmax and your Km, they are both changing in this case. This is an uninhibited enzyme, and Vmax is uninhibited here, and your Km. But if you increase the concentration, slope is increased. Again, the slope is increased. Oh, sorry, slope remains same, but your Vmax is increasing. That is decreasing, going up and up. 1 by Vmax is increasing, and this is going also further. So the three reversible inhibitors, first was competitive, in which your Vmax remain constant, but Km is changing. In the non-competitive, your Km is constant, but Vmax is changing. And in the uncompetitive, your both Vmax and Km, they are both changing. So that's the main difference between them. Mm-hmm. Now let's discuss about allosteric enzymes. So allosteric enzymes are those having other shapes or conformations induced by the binding of modulators. So these enzymes have two receptor sites. One site fits the substrate like other enzymes. Another site fits in the inhibitor or the activator molecule. And allosteric enzymes are very important in the feedback regulation. So here the inhibitor, they tend to bind over the allosteric site, and which will make your active site to be inactivated. But on the other side, the substrate is bonding to the active site. Of on the allosteric site, your inhibitor is not bound. So there are three types within the allosteric modulation of enzyme. That is allosteric activator and allosteric inhibition. So allosteric activator here, which give rise to a high activity affinity to the enzyme and will produce lot of products. And this active site will attach to substrate, and it will give less product. On the other case, in allosteric inhibition is low activity and little or no product formation is done in this case. And how is the kinetics of allosteric enzyme is? Basically, it will not give a hyperbolic structure, but a sigmoid curve here. So allosteric enzymes can be regulated between very low and very high reaction rates with only small changes in the substrate concentration. So allosteric enzymes are used by cells to regulate metabolic pathways where concentration of cellular substrates fluctuate over narrow concentration ranges. So allosteric enzymes show relationship between V0 and S that differ from the Michaelis-Menten behavior. And they exhibit saturation with the substrate when substrate is sufficiently high, and when V0 is plotted against a sigmoid saturation curve, which result in this. So the symbol S0 0.5 or K 0.5 we usually represent the substrate concentration giving half maximum velocity. The same thing, end product allosteric inhibition. So this is your allosteric site, and your end product allosteric factor is here, and this is active site substrate. So two things are possible. If the inhibitor attached, so substrate cannot attach. If inhibitor does not attach, then reaction will proceed. So some properties they have, allosteric enzymes have one or more allosteric sites, and they can bind binding sites distinct from enzyme activity site or substrate binding site. And then molecule that binds to allosteric sites are called effector or modulator. And effector may be positive or negative, and this effector regulates the enzyme activity. The enzyme activity is increased when the positive allosteric factor binds at all allosteric known as the activator site. And the binding to allosteric sites alter the activity of the enzyme, and this is called cooperative binding and allosteric enzyme display. So model plot of V0 versus S. So that's it from the enzyme kinetics. Yeah. So we are done, I think, with the kinetics of enzymes today, finally. And enzyme part is over. Uh, so this we have done all the introduction, amino acids, classification, structure of DNA, lipids, nucleotides, nucleoside biosynthesis, um, enzyme structure, enzyme nomenclature, introduction. So basically enzymes part is over. Now we can switch to another part. Metabolic and hormonal disturbances. Yeah. But I want to start this part to be on separate day, because this will take this is totally like your liver function test, your jaundice, hypothyroidism. So their all topics are together, and I want this together to be on separate day, not today. So we switch this to tomorrow then. And we are also done with all the virtual labs, I guess, also. Amino acids, lipids, enzyme, nucleic acids, virtual labs. So this is almost things are over. Now we have to switch to new and more exciting topics. So we will do this all tomorrow, students. For today, we are done at the moment, and we will start with the other things tomorrow then. Yeah. So thank you so much. Today is that's it. I want to just concentrate with the kinetics of enzyme concentration, go through them, revise them again. It's a bit complex to understand. Tomorrow we will start with more medicinal biochemistry, metabolic and hormonal imbalances like liver function test, your kidney function test, KFT, further hormonal disturbances and hyperthyroidism. Yeah, so we'll do some interesting topic tomorrow. Until then, thank you very much. I could start the topic now, but it will not make sense to like having different products to make the lecture then. Okay then, wish you a nice day. Take care. Bye bye. Yeah.